Hello everyone. I'd like to welcome you all in to the Revision Vault. Today, we're going to be covering part 1 of gas exchange, which is part of the AS Level syllabus for biology. Many people can find this a challenging topic, but I believe that it is actually one of the most simple topics within that year, so I thought I would break it down for you. Today, we're going to be covering mainly the trachea, but we will start off with the structure of the human gas exchange system. So here the syllabus requires us to know a simple version of what is actually inside of us. If you imagine every breath that we take, there are so many other functions happening within those few seconds. And the main places that these things occur is within the lungs, which we can see here on this image. The trachea, which is this large windpipe, which you may have heard also being called the airway or the windpipe. We have the bronchi, which are these smaller looking windpipes, and they break off from the trachea. There are two, each one going into each lung. We have bronchioles, which again are a smaller version of the trachea, and they break off into the lung structure. So that's why I've had to draw it on here, but you can also see it in this diagram. This is a terminal bronchiole, which is one of the smaller ones, and they are the final bronchioles because they lead into the alveoli, which are these grape-like looking bunches. So these are actually empty sacs, and they are where the main gas exchange occurs. And that is why we also have the capillary network, which can be seen here on this diagram. And these surround the alveoli. And that is exactly where our oxygen is transferred into our blood and carbon dioxide is given out. But we'll be going over that in more detail in other videos. So starting off today, we're looking at the trachea, which, as I've already covered, is this large windpipe with rings of cartilage around it. Here we can see a cross-section of the trachea, and this is where it's really important that we understand the different parts of what is going on inside the trachea. So here we have this ring-shaped cartilage, and this is the distinctive feature of the trachea. Does anyone know what this pipe is here? Well, you'd be right if you said it was the esophagus. And that is why we also have this break in the cartilage. I'll be going in that more detail shortly. We can identify the cartilage here on photomicrographs or electron micrographs, as we can see. And these polka dot type shapes is what is distinctive for the cartilage. Now let's go over those parts in some more detail. The cartilage is really important because it stops the trachea from collapsing. If you imagine a straw, when you suck up liquid in the straw, sometimes you can see the edges of the straw cave in. And that's because the pressure decreases as you suck the air up. And that is exactly what happens when we breathe in. So as air enters our lungs and goes down our trachea, the air pressure is really low. And this could cause the trachea to collapse if we did not have the cartilage. That is why it keeps the airway open and provides low air resistance, which then allows the air to travel through the windpipe without using too much energy. It's C-shaped, as I've already mentioned, and this shape is only present in the trachea, not anywhere else within the gas exchange system. The cartilage contains chondrocytes, which are those polka dots that I've shown you earlier, and we can see them here as well. They usually come in pairs, and that is how you identify the cartilage when you look at photomicrographs. Moving on, we have the smooth muscle. And as you can see on this photo, the cells are really elongated, and so are the nuclei. This has a really important role with relaxing and contracting, the same way your muscles do in your arms, your legs, anywhere else in your body. So when they relax, the airway widens, and this is often done during exercise to allow more oxygen to enter our system. When it contracts, the airway constricts, and this is usually in response to irritants, such as if we were breathing in a lot of smoke at that time. If you have asthma, or you know someone with asthma, then this is caused by overactive smooth muscle. Can you think why? Well, it's because if the smooth muscle contracts consistently, then that leads to bronchoconstriction, which means the airway is smaller than it should be, and that is why you get wheezing. As I've mentioned earlier, the smooth muscle is in the break within the cartilage. So when you have the cartilage as a C shape, the smooth muscle is present joining those two ends together. And that is because the esophagus is right next to it. And when the esophagus has food going down it, it carries out peristalsis, which is this wave-like motion, where again it contracts and relaxes to push down the food. And so it will expand in the future, which means that this smooth muscle must allow the esophagus to expand. If the cartilage was there, it would be too hard and the esophagus wouldn't be able to expand, which means we couldn't swallow food properly. On to collagen and elastic fibers. So within the trachea, elastic fibers and collagen are present in many layers. You don't need to know the details of this, but as you can see here, we have it in the lamina propria, the submucosa, the posterior wall, which is this area here, between the cartilage rings on the outside, and in the outermost layer, the adventitia, this one here. So elastic fibers and collagen don't have the biggest role within the trachea, but they do recoil passively during exhalation, which then reduces the energy needed to push out the air. And they stretch again during inhalation. But this is most prominent within the alveoli, and they have their most important role there. Moving on to the mucus. So we have a consistent layer of mucus coating the inside of our throat, which is also known as the lumen. This mucus traps pathogens, dust, bacteria, and other harmful particles, and prevents them from reaching delicate tissues in the lungs. These delicate tissues are mainly the alveoli. And that is why, as you go further down the gas exchange system, you will get less and less mucus present, because within the alveoli, we no longer want any risk of infection. It also allows the respiratory tract to keep moist and hydrated. And this allows gases to dissolve and diffuse across cell membranes much more easily. Mucus contains lysozymes too. And lysozymes are antibacterial, so they make sure that any bacteria that enters through our gas exchange system will be killed. So how do we get this mucus then? Well, most of the time it comes from goblet cells, which we'll be looking at later, but sometimes it is supplemented by mucus glands. So this is often when our respiratory tract is irritated through smoke or other particles, or even when we're ill. Mucus glands are multicellular. They're present in the submucosal layer, and they secrete mucus up towards the epithelium that you can see here. So this is the top layer. This is the inside of our respiratory tract. So he's sort of done a cross section, and this is the layer of mucus traveling above, and it comes up from the mucus gland. But as I mentioned, it's not always coming from mucus glands. It's mainly coming from goblet cells. Now, goblet cells have a step-by-step process of the way they create mucus and release it into our respiratory tract. So mucus is made of the glycoprotein, which, if you don't know what that is a protein plus a carbohydrate. And if you have already covered cell structure, which should be the first topic you cover in this year, then you'll know which parts of the cell could potentially be involved in making mucin. So you'd be right if you're thinking that rough endoplasmic reticulum is involved, because this is what synthesizes the protein that makes up mucin. And that is why when you look at goblet cells, you will see that they have a lot of rough ER, because you are needing to produce this protein. Then this protein moves to the Golgi apparatus in a vesicle, and that is where the carbohydrate chains are added, which is known as glycosylation. And it's packed into a membrane-bound secretory vesicle again. And additionally, there will be a lot of Golgi apparatus because of the reason of needing to modify this protein. Then the vesicles move towards the top surface of the cell, which we can see here. If you look at this vesicle, it is moving to the top surface and it is about to be secreted into the respiratory tract. And that is why we have exocytosis, which is an active process where the vesicle fuses with the membrane and becomes part of the cell plasma, which you know is that phospholipid bilayer, and it releases that mucus. So if you imagine, it joins up with the cell membrane, and this mucin that was inside is released into the respiratory tract. And this causes expansion because the mucin was condensed within the cell, and as it comes out, it expands to form that mucus layer. Well, how is this mucus then moved so that it's not constantly irritating us? Because mucus is the best place for bacteria to grow, right? It's wet, it's moist, it's warm, but we don't want these conditions to grow bacteria within our trachea or the rest of our gas exchange system. That is why we have cilia. Now, cilia move mucus up towards the throat. So the mucus is moved up this way, and that allows for it to be swallowed, which is what we do most of the time. But if you are ill and you have excessive mucus production, that is why then you end up coughing up the mucus, because it's too much for you to swallow. In order for the mucus to be moved up, the cilia must beat in synchrony, and this action requires ATP. That is why, when you look at ciliated epithelial cells, they often have a lot of mitochondria, because mitochondria produces the ATP needed for this ciliary action. Now, in case Cambridge asks you, they are 5 to 10 micrometers long. For some reason, Cambridge does quite like to ask about measurements, so make sure you're keeping track of measurements throughout this course. And we can see in this diagram here that the cilia are wafting the mucus up towards the throat. And lastly, we're looking at ciliated epithelium, because there are lots of different types. And as you'll discover later on in this topic, the different parts of the gas exchange system have different epithelial tissues. But the one that is present in the trachea is pseudostratified ciliated columnar epithelium. Now, I know that may sound complicated, but don't worry, because right here we have the diagram of it. And here is simple columnar. So we can see, first of all, the difference between pseudostratified and simple. Columnar simply just means exactly what you'd imagine, a column. So it's column-like with the cilia on the top and the nucleus within it. However, when you have pseudostratified columnar, they're sort of overlapping and irregularly shaped, with the nuclei at different levels. And so even though it may appear that there may be more than one layer, it is just a single layer. And we can see over here, it may look like some kind of fairy mushroom kingdom, but this is actually what is inside all of us. We have our mucus lining, we have our goblet cell, which supplies that mucus lining, and we have the ciliated cell, which makes sure that the mucus is constantly moving up to the throat where it needs to go. And all these other cells is nothing that you need to worry about at this stage, but I'm sure if you continue further in your studies, you'll find out what they're about. Thank you for watching today. I hope that it made sense and that you understood this topic. If you need any more answered questions, feel free to put them in the comments. Don't be ashamed of anything, because I want to make sure that you understand this part of the topic. This is the first video of my channel, so I thought I should introduce myself. For those of you who don't know me, my name is Sylvia, and here we're going to be digesting the CIE syllabuses for maths and sciences. From personal experience, I know it can be challenging when teachers struggle to explain personalized information for you. So here, I'm going to be trying to simplify everything that I can to build your fundamental understanding on these topics. Starting off,